I'm Dan Stanton, the editor and founder of BioProcess Insider. Astrea Bioseparations, until recently known as Prometic Bioseparations, is a supplier and developer of affinity chromatography and bioseparation solutions. The firm provides services for ligand design, adsorbent development, large-scale adsorbent manufacture, and GMP-ready pre-packed columns. And I am delighted to be joined by the CEO, Steve Burton. Steve, welcome.
Thank you, Dan. It's a pleasure to speak to you today.
Now, as I alluded to in the introduction, Astrea, until a few months ago, 6 months back, was known as Prometic Bioseparations. You were acquired by KKR late last year and integrated within KKR's life science tool platform, Gamma Biosciences, at the beginning of this year. So, Steve, I wonder if we can start by you explaining how this acquisition has kind of affected your day-to-day operations and, and your company's growth strategy as well.
Sure. Yeah, the company has been in existence for over 30 years. We originally were founded as a spin-out from Cambridge University, and for a number of years made our way as a small independent company. And then we, for various reasons, we became a part of the Prometic group. And for many years, I think we were a combination of a tools developer for the sale and supply of those tools to third parties, to the market, the bioseparations market. But also we were in part part of the internal development team for Prometic. So we were directly involved in the development, particularly of the protein therapeutic products as well. But as time went on, I think it became clearer that really we would fare better as an organization focused on bioseparations. Now that was, I think, our main focus. And that the time was right to separate the business from Prometic. And indeed, at the same time, Prometic itself was going through some changes as well. So Prometic today is known as Liminal. We, as a part of that sort of new thinking, at the beginning of last year, so beginning of 2019, started a process to structure the business for selling on, basically. And we went through quite an extensive divestment process that was managed by Lazard, and it was quite successful. So at the end of that divestment process, where we were pitching the company to multiple interested parties, we eventually were acquired by Gamma Bio, and as you said, a subsidiary of KKR. So we're very pleased with this as the new owner for the business, because we now have a very clear focus on developing and growing the business and its products and its clients in the biomanufacturing space. Whereas before we had a somewhat dual focus for the business, partly developing products for the market generally, but also being an integral part of a pharmaceutical development company. So that gave us some sort of being pulled in two directions, I would say, on occasion. So now we have a very clear focus.
So, as you say, the divestments, the streamlining, and the ability to really focus on what you do well has allowed you to move forward in your strategy. With KKR and with Gamma, are you privy to more increased resources, or are you looking to expand further on the back of the KKR acquisition?
Well, we're certainly on an expansion track right now. Being a part of KKR Gamma, this is a private equity company. So obviously they very much want to grow the business, to develop increase the value of the company going forward. And the way of achieving that, because it's essentially an equity growth fund, is to expand the business. So both organically by the further development and sale of our traditional products, but also by acquisition, so by M&A as well. So we're now very much looking at the acquisition of businesses which are aligned with our own to give us more breadth and depth in the biomanufacturing space. So Gamma Bio is about acquiring and growing businesses in the biomanufacturing space to become a significant player in the marketplace. And we are very much a part of that.
An exciting time. So as well as being focused on expanding as we go forward, I'm wondering if there are any opportunities to leverage some of the other elements from the companies within the Gamma Biosciences portfolio for Astrea.
There are, yes. At the moment, it's very much in its infancy. Gamma Bio, it's just starting out. As I said, we were the first company to be acquired, but there are other ones. So Gamma's recently announced an investment deal in the Univercells, and so they become the second company which are a part of the Gamma Bio group. Univercells is focused on in the upstream area, that they have disposable bioreactor technology. So we're in the downstream area. We have sort of the leading affinity technologies, downstream process technologies. So you can see from those first two investments and acquisitions in our case by Gamma Bio, how that group is going to evolve. It's going to become a leading bioseparations, biomanufacturing tools company.
Really complementary offerings there, and I'm very much look forward to hearing what else comes out of the what comes through on the Gamma Biosciences portfolio side of things. Steve, I'm going to shift gears slightly, mainly because you've been at the company and you've been within the industry for over 30 years. So I really want to tap you for your knowledge to see what sort of major shifts you've seen within the biomanufacturing industry during this period. I mean, obviously a lot has changed, but what really stands out for you?
I think you're quite correct. There have been a number of quite monumental changes in the industry, looking back over the 30 odd years that I've been involved with it. But in some respects, many of the original challenges remain, but in a new context. So I recall when I first started in the industry, it was very much a sense of the pioneering spirit. Mankind, scientists, technicians were doing things that had never been done before. And at that time, the focus was on producing proteins synthetically. So using genetic engineering to have cells able to produce human proteins in non-human cell lines, so in bacteria, for example, or in mammalian cells. And the focus initially was very much on the upstream side. How do we engineer cells to produce these protein products? The extraction of those protein products, the purification and the turning those into a GMP quality product that is fit for administration to humans was initially almost a secondary thought. The focus initially was on how do we make these materials in cells, and then how do we then make a product out of it? And productizing the biotechnology products, it wasn't a very quick process. It's something that has continued to evolve over 30, 40 years, and is still evolving. It's not ended yet, this process. So recently we've seen changes such as continuous manufacturing, continuous processing in the downstream side, where instead of making large single batches, there's now a move towards making smaller batches, but in a almost continuous mode. So they're run back to back very quickly. And that gives advantages in terms of productivity, savings in terms of space, for example. One doesn't need such large facilities. So, and very much a focus on productivity, that this is really where the industry has been focused in recent times, producing more valuable product in a shorter space of time at a lower cost. And those are, I think, are the main drivers for certainly the downstream side. And equally on the upstream side, we've seen big changes, again, in relation to the disposables, for example. When the industry first started, it was very much a stainless steel business, large tanks, pipes, fixed stainless steel systems. These days, the very large facilities still have that, and there are still some compelling arguments for very large scale manufacture for large fixed facilities. But certainly for the CDMO business and producing smaller quantities for clinical trials and small scale manufacture, there's very definitely an argument now towards a shift to these almost disposable systems. So we've seen that with bioreactors, disposable bioreactors. We've seen it with in replacing tanks with large bags, for example. So these have all been changes that have occurred over a number of years, and this is still evolving. But now the industry is not moving away. It is focusing on new therapeutic agents. So the protein business is still there and it's still growing. But there are now new therapeutic entities which are being looked at in detail, and particularly in the cell and gene therapy space. So where we now have viral vectors as therapeutic agents and DNA as a therapeutic agent. So these are relatively new things in the industry as a complement in the traditional proteins. And one can see a similar kind of trend to the way that the protein biotechnology industry first started. So today we have quite a lot of focus on the upstream side. How are these vectors going to be produced? How do we engineer cells to produce these viral vectors? And how do we deliver the DNA payloads into cells? So that's very much a focus of the cell and gene therapy area today. But we then have this growing understanding and realization that these are on the track to become therapeutic products in their own right. And in fact, there have now been, I think, 3 or 4 cell and gene therapy products now approved for use. So early days yet, there are many hundreds, probably thousands of cell and gene therapy products in development right now. So it has striking similarities with the beginnings, the early days of the protein biotherapy market, with the focus being on the upstream and this progressive realization that there needs to be much more focus on the downstream side and the productization side of these materials. There are materials, there are separation technologies used today for these products, but a number of those were not specifically developed for cell and gene therapy products specifically. They were developed for other applications, mainly proteins, and they have been adopted and used for these new therapies, but they're not always appropriate. And we're now seeing the development of separation materials, which are really geared to the needs of the cell and gene therapy industry. And so this is something that's happening right now, and it's a relatively new thing in the industry.
So what you're kind of saying is that we're seeing a lot of the issues that we had in the early days of antibodies and biologics coming through with these new modalities. But really, the next part of the puzzle, so to speak, will be the downstream. Now, that leads us neatly to Astrea's space part in the industry. What are you doing at Astrea to overcome these challenges with these new modalities?
Well, our history is certainly in the area of affinity separations, and affinity chromatography, certainly over the years that I've been involved in the industry, has really come to the fore in the downstream field. So if one looks at antibodies, for example, monoclonal antibodies, Protein A has really established itself as a true platform for the manufacture of monoclonal antibodies. And monoclonal antibodies today represent over 50% of the market for biologics, and still growing. The majority of those antibodies are purified with one affinity adsorbent, with protein A, one affinity ligand, should I say. And there's been huge investment over the years in that ligand to really maximize its performance for antibodies. And as a consequence, it's really changed the way that proteins are manufactured. So today, if one looks at what would be an ideal manufacturing process for proteins, it would start with an affinity capture step. And that was not the case many years ago. It will be a question of combining together various separation modalities in such a way that one achieve the desired purity for the target and maximize the yield. But of course, the larger the number of individual steps, the greater the losses and the lower the yield is going to be. So having a very efficient capture step right at the front of the process is a way of maximizing the yield. It's a way of simplifying the process, because then the subsequent steps are really polishing steps rather than further capture purification steps. And the cost of goods has come down dramatically as a consequence. So while the antibodies remain the focus of the industry, the focus was on protein A and how to improve protein A. Now, during this period, we were looking at other ways of developing affinity ligands. And we've developed a number of techniques which really have their origins in the small molecule drug discovery space. So, because what the objective is to have a selective binding ligand that's able to recognize and bind to a certain region on the surface of a protein or a virus or any other biological entity, and bind that entity in a selective fashion. So it binds it to the exclusion of other compounds that may be present, and then they can be removed by washing away. When one thinks about it, drug discovery, drugs work in an almost similar way in the context of the binding, but it's inverted. So most drugs are soluble entities that are focused and bind very specifically to immobilized receptors, so typically proteins receptors on the surface of the cell. So there are very many drugs that work in this way. Affinity chromatography is almost the inverse. We have a soluble receptor and we have an insoluble, immobile binding entity. So we call it a ligand, not a drug, because it has no therapeutic effect. It is sole focus, the sole objective is to have selective binding to the target, but it has no other functional activity. So the techniques that have been developed for small molecule drug discovery are equally applicable to the discovery and development of small molecule affinity ligands. So, for example, we can use high throughput screening. So we can make libraries of potential affinity ligands. We make those libraries of compounds using small organic molecules, which are attached directly to the surface of chromatography beads. And then we can screen these libraries en masse, basically. So we can make and test relatively large numbers of compounds quite quickly to determine if any of those have binding activity for the therapeutic entity that we're interested in. So one needs alongside that to have assays that are able to detect the target and distinguish it from the other impurities. So that's one way of doing this, and this is like a mass screening approach. And there are a variety of different ligand platforms that can be used with this approach. So we have used extensively these small molecule triazine-based ligands. We use triazine chemistry because it's very controllable. It's been very successful in the development of affinity ligands. It has its origins way back in the some of the earliest synthetic ligands that were used for protein purification were actually dyestuffs. Now we don't use dyestuffs today, with one or two exceptions, but the linking chemistry that was used is proved to be very useful for these modern versions of those original sort of synthetic ligands. So one can use this this screening approach that relies on really on serendipity, that there's a compound in the library that just happens to have the right shape, the right charge distribution to bind to the molecule that we're interested in, and doesn't interact with the compounds that we're not interested in, so the impurities. That's one way of doing this. There are other ways of doing this. So increasingly, we're now turning to another drug discovery approach, which is computational chemistry. So these days, one can now visualize the surface of a protein or a virus using the structural information that's commonly available these days on these entities, so X-ray crystallography, for example. And then interrogate the surface of that biological molecule and look for potential binding sites. And there's various ways that we can do this. We can also, having identified potential sites, we can envisage molecular structures that will be able to fit in that site, that will be able to interact with that site in a certain way. So we can now perform what are called docking procedures using docking algorithms. So we can dock small molecules into sites on a protein and measure, gain a measure of how effective their binding would be, typically by looking at the change in free energy associated with binding. And we can also do virtual screening. So we can now actually have a virtual library of potential ligands. So this is not a real library that was synthesized. It's a library that exists electronically with descriptors of the chemical descriptors of the ligands in that library. And then we can use these computational packages to essentially roam these molecules over the surface of a target and identify potential binding sites. So we can do screening in a virtual fashion. And that enables us to screen many, very large numbers of compounds relatively quickly and gain an insight into the types of molecules that might be effective in binding to certain regions of the target. And at that point, we can then make our compounds, our libraries, and screen them. But now we're making more focused, more targeted libraries that have a much higher chance of interacting with the target at that site. So we've removed this serendipity now from the equation. We're now starting, rather than with a very large random library and relying just on pure chance that a molecule in there happens to bind the target in a way that we want it to bind, we can use computational chemistry to predict the sorts of compounds that will do that. And then we can make much more focused targeted libraries and perform the actual screening on those. So when we first started, I mean, even our basic libraries, we could potentially make millions of compounds. In the early days, we certainly used to make thousands, tens of thousands of compounds, and we would screen those. These days, we would make libraries of maybe a couple of hundred compounds that we've identified from these virtual screening techniques, and we will screen those, and we're seeing actually successes that are probably greater than we would have had previously. So it's definitely an advantage, and it's speeding up and simplifying this whole discovery and development of affinity ligands. So it's quite an exciting environment to be in, to be able to very quickly discover new binding ligands using these modern design techniques.
And possibly very crucially, customizing the specific ligand for the specific therapy. Steve, very quickly, how does that translate to the cost of the manufacturing process? If you can hone in and choose a specific customizable ligand rather than go for the very expensive protein A?
Yeah, actually, I'm glad you raised that point, because it is quite an important one. A key reason why we have focused on these small molecule ligands, apart from our ability to develop them to bind to almost any target, is the cost and the utility of these small molecules. Selectivity is important, and we've certainly had very spectacular selectivities, in terms of their ability to bind one protein, approaching the selectivity of monoclonal antibodies. But one mustn't forget, these are small chemical entities. So rather than using, if one was to use, for example, an antibody, and antibodies have been used as affinity ligands, and there are many examples of that, but those antibodies are derived from a biotechnology process themselves. They are effectively monoclonal antibodies that have been produced in a bioreactor. They have to be purified themselves before they can be attached to a chromatography support and used for purification. So that comes at a price. They have very high binding avidities. Sometimes they bind the targets too tightly, but they come at a cost. Now, for small scale purifications, that can be an issue. For large scale purifications, it can also be an issue in terms of the availability of the material. Now, going back to protein A, where there's been so much effort placed on the development of protein A, and it's become the standard ligand for monoclonal antibodies, the cost of developing protein A was more than offset by the numbers of users, the market demand for protein A. But if we have other therapeutic targets that are not antibodies, where the market may be much smaller than the market for antibodies, then the cost of the ligand becomes a larger issue. And certainly the cost of developing a molecule like protein A would be seen to be very expensive in comparison to a small run for an initial manufacturing campaign for a new therapeutic entity, because the whole cost of developing that ligand is then focused on that one application. So to overcome that, the small molecule ligands that we have are much less expensive to produce. We're using traditional chemistry to synthesize these, rather than the biotechnology techniques. And even compounds like peptides, I mean, we also have peptide libraries as well as the trizine libraries, but they can become themselves quite expensive to make and quite limiting in terms of scale. And we certainly would only produce peptides up to around hexamers, or around 6 amino acid long. Beyond that, it becomes quite expensive to produce them. And just to put this in context, a typical affinity adsorbent may have in the region of 10 mg per mil, so 10 grams per liter of affinity ligand attached to it. If we look at a scaled up process that might have 100 liters or 1,000 liters of adsorbent potentially, then we're not talking about milligrams or even grams of ligand. We're talking about kilograms of ligand. And that needs to be understood. Now, with these small molecule ligands, we can easily make kilogram quantities of ligands. We do this on a regular basis. To make kilograms of a recombinant protein ligand is a much more challenging and much more expensive task. And that actually limits the scale at which these can be used at. So you're quite right to raise that the cost, the availability of these ligands, and also the utility of them. I mean, anything that's derived from a biological system is likely to be biodegradable by its very nature. So by having a synthetic chemical ligand, we can engineer these so that they are essentially non-biodegradable. So we have ligands in our libraries that we can autoclave, we can heat treat if necessary. They can certainly be treated to high concentrations of cleaning agents like sodium hydroxide. So the majority of our ligands are resistant to even 1 molar sodium hydroxide over many, many cycles. I mean, we have had users use these adsorbents over 1,000 cycles because they're incredibly robust, and they can be cleaned with these very aggressive and very effective cleaning agents. And that combination makes the cost, the overall cost of using these synthetic ligands very low in comparison to using things such as monoclonal antibodies.
Could this be the death of the recombinant ligand?
I wouldn't say it was the death. I would say that these are two approaches that sit in together in parallel. Like all technologies, there are situations where some are better suited to some applications than others. So I think there will always be a requirement, a demand for the protein-based ligands. Certainly they have very high binding affinities and very high selectivities. So there may be situations where that is a desirable requirement. But equally, there are many other situations where, particularly where proteins are manufactured at large scale, where we're not talking about sort of grams of protein being produced. We're talking about tons of protein being produced. Plasma proteins are a good example of that, for example. So we have ligands that are used very effectively today for the isolation of albumin, the isolation of other plasma proteins, plasminogen, for example. And some of these are produced at very large scales, and they use very large columns to do this. So some of our products have been packed into columns 2 meters in diameter. So these are very large scale applications. And we can offer materials to clients that enable them to use them at these scales and for these types of manufacturing operations because of the relatively low cost of the adsorbents and these synthetic affinity ligands. So we have the best of all worlds. We have the selectivity, but we have the low cost of the ligand, and we have the ability to make large quantities of it relatively easily.
Well, Steve, I think we've gone way beyond our time slot, but I want you to take that as a testament to the quality of this discussion. Thank you so much for speaking with me today over this video call. It's a shame we can't be sitting in a exhibition hall in San Diego, but you know, there's always next year. Steve, Burton, thank you so much again.
Thank you.